Hello everyone. I hope you all can hear me. Please confirm in the chat, put in the comments that you can hear me clearly. Quickly, guys. Can you all hear me? Okay, that's great. Hello and welcome. My name is Dilukshi and I will be your host again this evening. Shekhar sir will be joining us for the remaining sessions. I hope you all remember we have 2 more information-packed sessions to go. Thank you all so much for joining us on day 4 of the 3D bioprinting webinar series powered by Altum Technologies and Biotechnica. So today's topic is bioprinting complex drug carriers for localized delivery. Many of y'all were already curious about this topic when the series started. I remember reading a lot of comments about drug designing and drug development. So as we go further into these sessions, you all will be learning more. Keep your ears and your eyes peeled because we will be going more in depth. So I hope you have all of your questions ready. So before we begin, I have a humble request to all of you. Kindly share this webinar link with your peers, your friends, and your colleagues so that we get a lot of eyes on this series. We require your constant support and encouragement to last for more sessions to come. Your support is what motivates companies like Altum and even us to organize more such industry-sponsored sessions for you. So I hope you all have shared the link. So some instructions to note before we begin. As usual, there will be a Q&A session. So kindly post your questions with the letter Q and then your question. I repeat, post your questions with the letter Q followed by your question. And as for feedback forms, the link will be provided in chat at the end of the webinar. And also stay tuned for all of the sessions and listen carefully because there is a bio quiz competition that is ongoing. So don't forget to check out the LinkedIn page of Altum. Okay, make sure you go check out the LinkedIn page of Altum, follow them there, answer a few simple questions, and you'll stand a chance to win an Amazon prize. So all instructions are provided on the Altum LinkedIn page. So without wasting any more time, let's welcome our speaker for today, Miss Aishwarya, who's back to give you more insight. I hope you all are very eager to learn.
Hello everyone. I hope you can hear me.
Hi, Aishwarya.
Dilukshi? Yeah,
Yes.
hi. How are you? Yeah.
I'm great. How are you?
Yes, I'm great. So I'll begin with the session.
Yes, sure. The stage is all
All right.
yours.
All right. So, good evening, everyone, and thank you for joining me on this session, which is our 4th session from Altum Technologies. The topic for today is bioprinting complex drug carriers for localized delivery. So, before I get to this topic that we have today, I just want to just recap through our previous sessions to give you a more flow continuity to this. So I think on our first session, we talked about what is bioprinting, the applications in tissue engineering. Followed by that, we spoke about soft robotics, and then we talked about microfluidic devices. And then my fellow colleague Divya has spoken to you about 3D cell culture platforms. And now we're coming down to drug delivery platforms. So, just introducing myself again, I'm Aishwarya Shirur. I'm a technical specialist at Altum Technologies, and patiently wait with me through this journey, and I will explain more. And through all of these sessions, you will understand better and better about this technology and how you can use it for your specific projects or your ideas. So, talking about us, Altum Technologies, we are a 3D innovation platform company, and we have a lot of partnerships, or rather, we work with multiple products in the industry, say Dassault systems for CATIA. Then we talk about MSc software for simulations, say computational modeling, other sorts of simulations, and Stratasys for their plastic printers, the 3D printers, followed by Artec 3D. And these are into 3D scanning. And then we have our life science solutions, which is BioVia for life science simulations, and CELLINK for 3D bioprinting. So, talking about Altum, we started around 2011, and we have been setting our foot in the industry with couple of awards, with our partnerships with people throughout, and that's how we've been growing, and we hope to grow further. Um, Along with this, I mean, I understand that most of you are from the maybe the life science background or the bio background, but I would also urge for people to pass on this message to the people in different industries, say automotive industry, automobile industry, say aerospace industry, or any other industry where you think these mechanical softwares, these life science softwares, these biovia softwares will come to use. So kindly spread the word, as well as follow us on Altum Technologies Private Limited on LinkedIn and other platforms. So I will. So this, obviously, this is our customers. We have several set of customers, and obviously growing set of customers in the education and research, aero and defense, automotive, healthcare, then obviously the consumer industry, and the and several other OEMs. So we've been growing in different areas, and our idea is to provide you the right solution to anything that completely brings in creating a product or any solution to a problem. So while we come to the main topic, which is again drug delivery, I will just start with the basics on what is the whole system of drugs, how do they travel into our body, how does they go about, what is the kind of requirement, or what are the things we need to consider. Following this, we'll obviously go about how we can incorporate the technology of bioprinting into it, and how we can make this a little more efficient in the existing methods. So, as you know, drugs are pharmaceutical substances, and also called as maybe medicines, or a medication. So, it is a chemical substance which is used to treat, cure, prevent, or rather diagnose any disease. And so, the idea behind using any drug is obviously to promote well being. And when you talk about drugs, they are supposed to be a treatment to any specific symptom or any main ailment. So always there arises a question that we have had a drug, what happens to this, right? What happens to the drug in your body? So when you administer your drug, so the first step is going to be the drug administration, or rather the dose of drug administration, right? So this is where they are going to give in the drug to you, and this drug administration rather comes in different dosage forms, different doses. Example, you go with mgs or ml. So these are obviously calculated, and there's a whole different calculation under, or the whole different process in getting to it. From this dose calculation and administration, we go into dose concentration in the systemic circulation. Obviously, this is where you have consumed your drug, and now it is going to your body, right? It's going through your body and going through the target site. So before it goes to the target site, you actually have your drug metabolism, which happens. So you need to, in certain cases, not all cases, in certain cases, drugs need to be metabolized, or rather broken down from their existing structure to a different structure, maybe a more simpler structure, or rather it has to be even eliminated. So once this drug has passed through your body, there is also the step where you have, say, metabolism or excretion. Simultaneously, it also needs to go to the site. It needs to go to that specific site where you want it to act upon, right? So that's where you have the drug which reaches your tissue, and that is called distribution. So when you start the process of taking the dose concentration into your body, it is called absorption. And then following that, you have metabolism, or metabolized drug. Then you have elimination, and then you have distribution. So this is how it kind of comes into the area of ADME studies, if you've heard about it. So as this whole process happens, you also have drug which comes at the site of action, followed by the pharmacological effect of this drug. So this is actually the sequence of events that rather happen when you talk about drugs, from the time you take it to the time it acts upon your body. So pharmacokinetics, right? So this whole section where the drug is moving throughout your body is a study called as pharmacokinetics, and the part where how the drug acts, right? How is the drug interacting? How is it giving you a pharmacological effect? How are you responding to the drug, and how are you how is your fever or are there any other problem subsidizing? So that's where you have pharmacodynamics. So this is the whole crux of how drugs are and how drugs act. Honestly, this is just the basics of it. So as we proceed, we have, right, this part where you have dose of drug administered. The dose of the drug administered would also be in the form of drug delivery systems. Right, so drug delivery system is actually a method or process of administering a pharmaceutical compound, and the idea is to obviously achieve a therapeutic effect in the humans or in the animals. And when you talk about drug delivery systems or any form of drug delivery, we need to know that the drug that is released, drug release is basically it coming to action, or it transporting or breaking through its encapsulated layers in a more controlled, slow, and targeted manner. And obviously, sustainable release is another main point that you need to consider. So, drug delivery systems obviously is an engineered technology for targeted delivery and for the control release or release of therapeutic agents. So, when you talk about the conventional systems, we have the buccal, oral, pulmonary, transdermal, ocular, nasal, and obviously the suppositories, the vaginal or anal form of delivering the drug to your site. So these are actually the conventional routes by which your drug is actually delivered. And when you talk about coming down to something else, so when you have your drugs, right, these conventional methods, they have a problem in them. The problem is that sometimes, I would say sometimes, not for all the drugs, they would not reach the specific site efficiently, or rather there's a lot of side effects, or there is less amount of availability of the drug at the site. So as you know, it, say example, you're talking about a tablet, it passes through your entire GI tract. It is obviously going through this whole process of digestion that as we talk about it, where there is acids and alkaline solutions which are going through the medicines, and obviously this is going to reduce the bioavailability of the drug. So that's where we have to play around with drug dosage, the amount of drug you're going to administer versus how much is going to really affect the body of the person to be therapeutic enough, right? So when you talk about these drug delivery systems, they had conventional methods, and then they obviously came down to novel drug delivery systems, or rather uh, you would call them as complex drug delivery systems, or rather smart delivery systems. So here the approach is actually the formulation, where you're transporting the pharmaceutical compound in the body, obviously in a more safer manner, and you would consider maybe targeted delivery, sustained and controlled delivery, and modulated delivery. So that's again the whole idea of us trying to get into complex or novel drug delivery systems. So as you can see in this picture, you have several areas that we're talking about, and ideally one of the main factors or one of the main sections when it comes to using drugs is the formulation of drugs. And when you're formulating drugs, you have nano carriers, and you have hydrogels and microparticles, and obviously cellular vehicles which can transport these medications to the site. And delivery routes are something where you have transdermal patch, any sort of gel, any sort of injection, right? So the obviously this other half is where you see the immunotherapeutic agents, and these are all something related, something more complex, to be honest. And obviously the section we can tap into, with vaccines. I remember everyone, please take your vaccines. And obviously coming back to our main idea, it's that we want these drugs to be site specific. They will have less side effects. They would have greater bioavailability. So whatever explained about the whole drug going through your body, there is some amount of wastage while it gets through your body. That's why you use the term bioavailability. So with drug delivery systems, now this is just an example which I would give. uh, you see that this is an example that they would use for ocular treatment, And there are a set of drugs, a set of complex and rather novel drugs that they're using. So you have microneedle delivery systems, and then you have microspheres, you have nanoparticles, you have implants. So the when you're talking about implants, they're supposed to be more rigid, they're supposed to be more mechanically stable when you're using it on a body. And then obviously you have drug-eluting stents, drug-eluting lenses. These are all few examples that I would like to give about our complex drug delivery systems. So as we transcend and see through all of these are obviously going through some conventional processes of creating these drug delivery systems, example microneedle or microspheres or even nanoparticles. I'll also give you maybe a few more examples as we proceed with this. You have the stent, right? This is obviously a um, conventional stent that we use in terms of any atherosclerosis. And this is where a stent would actually hold your entire vessel, your artery in place. And while the stent is there, it is supposed to allow the sufficient flow, and it also has this capability of eluting the drug. So now, when you have a simple structure like a stent, which we've already developed previously, and if you're able to create something more biocompatible and something more drug eluting and more sustained, it is going to be rather a very good or a great victory in the field of medicine, that we are transcending from any sort of thrombosis, morbidity, any sort of negative impact of drugs on the body, or rather eluting stents on the body. So we are trying to transcend from whatever side effects that we have been seeing with the drugs, and we want to proceed to eliminating those. So with NDDS, we obviously have many types, like say example liposomes and nanomaterials and microspheres and self-assembling ones. So there are a lot of examples. I will not get into depth of that because we have something more in detail to discuss. So these are those are the main ideas, right? So if you remember, I mentioned about hydrogels in this section. So this is where we kind of get the connection to how 3D bioprinting comes into picture. So when you talk about hydrogels, these are three-dimensional structures, and they are polymers obviously, and they have the capability of absorbing and retaining large amount of water. So because they can retain and absorb large amount of water, they are very what do you say, adaptive. They have the capability of staying in a certain type of environment to sustain and rather help in providing the sort of drug delivery that we need, right? So hydrogels are very tunable and very versatile materials. They go through a lot of fabrication process obviously, and they have been used a lot in our biomedical applications, quite a lot. If you talk about the types of hydrogels, we have our natural hydrogels, say hyaluronic acid, alginic acid, pectin, dextran, chitosan, collagen. And obviously you've heard of gelatin, fibrin, and gelma. So these are all natural examples, natural hydrogels, and then you have synthetic hydrogels, which is PEG, PLA, PLGA, PHB. So these are quite often heard of. These materials are quite often heard of, and there's a lot amount of work happening in terms of hydrogels and the usage of hydrogels in various sections of medicine. So when you talk about hydrogels, it's not very simple. The process of extraction, formulation, optimization, characterization, these are all very important words to consider, because that is what is going to help you create the final product. So when you're talking of this hydrogel, or example X hydrogel, um, you would consider various factors. For example, biodegradability, right? I need to know if my hydrogel that I'm using is biodegradable. It's able to degrade over time. Is it causing any sort of cytotoxicity to the cells? Is it causing any impairment? So we need to check that when you're considering a hydrogel. We have to check the ionic charge. So everything that we do, every cell that we have, the nucleus, the cell wall, everything has a specific charge. Say proteins have, say, positive or negative charge. Similarly, ionic charge is something which is important. So we try to consider what is the best charge that we could use, so that this will provide the right amount of efficiency when it is administered to your body or even used in general. So you have obviously other physical properties, say smart hydrogels or conventional hydrogels. So conventional hydrogels are probably just the hydrogel itself. For example, I've used just collagen to create something. But there's one thing about all of these hydrogels. They are mostly, at least when we're talking about natural hydrogels, they maybe lack mechanical or viscosity properties in them, and maybe they might lack a few characteristics that we need, because, for example, we're talking about cells being printed along with the hydrogels and we want it to be biocompatible. But there's also a factor that when you print it, it needs to have something called shape fidelity. It needs to have uh, the right amount of viscosity when you are going through the process of printing, or rather even creating something. The concentration of the polymers is quite essential when it comes to using it to go through the process of creating a final product. So smart hydrogels will be a combination of other materials along with your existing hydrogels. So example, I want to use alginate along with cellulose. So when I combine alginate is a very good material, a natural source of hydrogel. It is the best in terms of creating the environment for cells and creating the ECM for the cells. But it also lacks this capability of mechanical strength. That is where you kind of mix or you use another material, say cellulose or gelatin or gelma, to improve the viscosity and rather the strength of that. So I think the other parts of it are quite simple. What is the preparation method of it, etc. So with hydrogels, obviously we would go down to understanding what is the next way that you could use it. And I also want to stress, it's called a three-dimensional cross-linked polymer network. And the question always comes down to, okay, if it's a three-dimensional network, why would you need um, another 3D design model to create it? It's obvious that we are trying to create physiology. That's where 3D bioprinting comes into picture. So 3D bioprinting is a form of additive manufacturing, as we all know, and this uses cells and other biocompatible materials as inks. So this is to print living structures, and obviously this goes by layer process to mimic the behavior of natural living systems. So when you talk about 3D bioprinting, you can see this structure that we had shown quite often previously, where they try to print something like a ear structure, an artificial ear or anything of that sort. And when you come down to 3D bioprinting, it's actually the collaborative technology, rather you would say, where you have 3D printing, right? General plastic 3D printing, wherein you're printing layer by layer to create any specific design that you want. Along with that, we are involving tissue engineering concepts, and these concepts would be form, fit, function, repair, regeneration. So these are the ideas you use when you are using 3D printing, and that's where 3D bioprinting actually was born, when you try to integrate these two technologies into creating something that will make research in general easier. So there are main components, that is cells, bioinks, culture media, and like I said, cells are dependent on the source. The cell source could be, say, example, skin, keratinocytes, fibroblasts, and then you have your bioinks. In the bioinks, like I said, we spoke about hydrogel, and one of the main essence of a bioink is actually the hydrogel. A hydrogel is also called as a bioink, by the way. And this bioink, right, is also called a bioink sometimes, in some cases, where it is mixed with a good amount of cell suspension, right? So when you have a hydrogel plus a cell suspension in the ratio of 2 is to 1, then you have created something called a bioink. But even generally, bioinks are hydrogels which are optimized. So these are tweaked enough to be used for the sake of printing, right? That's why they have the name bioinks given to them. And obviously, you have culture media, which is necessary for the cells to sustain and grow in, and this gives that right amount of environment for the cells. So, with 3D bioprinting, obviously, I'll just give you a very simple example here. Right, so they're just doing the extrusion and they're trying to print a simple structure. So this is where you would use a set of print heads, to create multiple structures, maybe with multiple cells or maybe with multiple materials to create a more complex structure. I know that this looks like a very simple structure, but you can create more complex forms, which can obviously replicate the physiology or the anatomy of the humans. So, with this, I will now come down to the cases. So, there are quite a lot of interesting cases to talk about when it comes to the use of bioprinters in creating something. So, this case is actually the development of 3D printed drug eluting scaffolds. So, they are creating scaffolds now to prevent piercing infection. Okay, so I'll just give you a background on this, or a general. I mean, this is something that everybody knows or observes these days. Every time you get a piercing, you have this tendency of getting an infection, and this infection is obviously due to microbes, and obviously this causes a lot of problems later on if the infection spreads, or it sustains, and it might even spread throughout the year in some cases, or sometimes even leave a scar behind. And then obviously you need antibiotics and antimicrobial creams, etc., to apply in those sites, because that's the only way you can get rid of this uh, infection which is caused at that site. So this is actually a case wherein they have tried to print a biopierce. So biopierce is nothing but um, a more gel-like structure which is attached to your pierce stud, whatever stud you have that you're going to apply this that point. That's where they're trying to like create another additional coating of a medication to allow proper drug delivery. So in this case, they're using mupirocin. Okay, this is loaded with MLGA. So mupirocin is a very commonly used drug in the cases of any infection. So they are usually edible drugs. And as I told you, that our problem sustains that we want target delivery. We want that site which is infected to be easily recovered. So obviously the processes are going to be that you are using a bioink, rather a hydrogel, and here they have used PLGA material. So they have created this hollow cylinder, which is 8 mm and 1.6 inner diameters, right? So they have a specific dimension that they have set. You would obviously print whatever structure you want, keeping the speed in mind, um, keeping the nozzle's dimension in mind, because I'm creating something as small as say 1 mm, and we are trying to make it more concise and more effective. So these kind of specifications are very essential when you're using a bioprinter, because we need to know how efficiently we'll be able to create that design that we want, right? So obviously, as we go through the process of printing a specific structure, you have, say, example, PLGA. So PLGA is actually dissolved in a specific type of solvent in order to allow better flowability or to allow the printing. And as we made the printing is, example, done, and we have to now go through a set of tests. So the drug which is loaded, obviously it's a mixture of PLGA and mupirocin. It is printed together and the structure is formed, and now we have to test quite a lot of things. So example, the solvent removal, whatever solvent we have added should not cause any bad effect. Next is the drug integrity. So the drug integrity, as in how long is the drug release happening, is a drug at the right concentration, is it giving me the right amount of efficacy. Then the drug release. Drug release, obviously, the drug will be in a specific encapsulated form. We need to make sure that it is sustainably uh, or efficiently going at the site, and the efficacy in other tests are done. So over a week, so the retention time of the mupirocin obviously was tested, and they found that this did not change. And now this happened from a duration of say day 0 until 2 weeks, that is day 14. So they went on checking and testing with different concentrations of the drug, with different concentrations of the hydrogels, and they try to see what is obviously the best concentration of the drug you could use, and also what can be the best integrity of the biopierce created. So we need mechanical strength, because when you're using a biopierce, you are also piercing this along with the hole that you created, the earlobe, for example. So this has to go along with that and has to stay at the site. So they try to check any sort of uh, mechanical damage that could happen to this biopierce by any chance. Also, you would see that there are no degradation products. So whenever a material or a drug, anything, basically any chemical compound, right? If it comes to a site, there's always a degradation that happens. It has to break down. It has to come affect the site and also break down. And while it breaks down, we have to make sure there are no byproducts which are bad to you. Basically, they're not supposed to be toxic. So we have to do a set of toxic studies. You check obviously through your scaffold tests, where, say, example, in this case, they take a microbial and they put down the scaffold. So the scaffolds are obviously created in various sections. I mean, you always have a test and you have the main drug which you're trying to test. So when you have your test drug versus your main drug, you would also see the something called a zone of inhibition. Whenever you have any microbial study that happens, you would try to place that drug at that site, and then you see what is the zone of inhibition. Basically, the zone of inhibition is till what extent is my drug elution happening? Is it sustaining over time? And what is the spread of this drug over there, right? So these are the kind of tests that they have to carry out to bring about something as simple as a biopierce. So from here, we have another example, which is a 3D printed microneedle patch. So this is a system for, say, pain removal. And when you talk about a microneedle patch, right, I had shown you in one of these slides. One second. Yeah. This is a microneedle patch, right? So the idea of microneedle patches are that they are supposed to be transdermal. They are supposed to penetrate through your skin and obviously reach that site where the drug is supposed to go. So in this case, in this study rather, they have tried to do the pain management using a microneedle patch. And I'll give you two cases or two essential factors that we need to consider, especially in this case. One thing is when you're talking about, say, example, lower back pain or any muscle pain, we generally tend to go with the lidocaine, gels or any type of solution which you can apply topically on that site, which can allow the pain to subside over time, right? And another thing is that when you are doing these things, these are um, ointments or rather other drugs which are obviously staying more superficial. So we are trying to improve the penetration of the drug into the specific site through the stratum corneum, which is the first layer of the epidermis. So as this drug starts to proceed and break through the barriers, right? So we are talking about small molecule drugs and larger molecule drugs. And as we go through comparing and understanding that we need to use drugs which are more efficacious, we would have to we would have to also break a certain rules or a certain barrier in terms of getting to that point. So when you're using microneedle, obviously this is to help our penetration. Another thing is when you're going through a back pain, again the same example, um, heat treatment is one of the essential sections, you would say, where it actually allows in relieving the pain, right? So what if you could create something which is integrated, a system that is integrating a microneedle patch along with a heat treatment system? And that's this case where you're actually using a transdermal patch, right? This is to diffuse the drug, or basically allow the diffusion of the drug, and also the heating of the drug. And the materials that are used here are PDMS, right? And carbon nanotubes. So PDMS, we know about silicon based materials which are widely used. With PDMS, they have added carbon nanotube. So carbon nanotubes are actually materials which are good conductors of electricity, right? So they have now tried to mix two materials, create a micro heater system. So micro heater system would be what they print themselves. They bioprinted themselves, and they would attach that to your micro needle patch. So you have your micro heaters in combination with your micro needles, which can efficiently allow the treatment of, in this case, right? So here the microneedle, obviously in the microheater needle accelerates the dissolving process of the drug, right? So when you have already punctured through the skin and you have put the drug, it has to pass through it, it has to diffuse to it rather. And as it is diffused, we also need to control the amount of diffusion, and the control of diffusion is happening through a microheater attached to sensors. And we would actually see throughout the study that they are trying to they're trying to see what is the subsequent amount of changes that can happen in the diffusion of the drug in the case of a model of a rat skin, right? So they see what is the diffusion of the drug, and they use a dye material to see what is the amount of penetration of the drug and to what extent the drug has reached the site. So here the mild heating, or rather an elevated heating, will increase the amount of drug diffusion over time. And obviously due to this, the thermal pressure that you apply here is also going to allow better amount of circulation of blood. So that these are the two factors that they are trying to play around with to create a microneedle patch. And now in this case, they have used a rat model. They have used it on the skin of the rat to see the penetration of the drug which has gone through it. And conventionally, when you bioprint these, you would obviously consider factors like speed, and the temperature. Then you have the nozzle dimensions that you're creating. You also have to see whether you're able to create the right resolution of what you print. So this is a needle that you're trying to print. Conventionally, when you try to create any sort of micro needle of this sort, you would go through molding process. So molding process is basically using a cast, using the material you want, make it print over the cast. Basically, there's a negative of the cast created. You print that structure you want, and then you have the peeled off peeled off part of the negative, which is going to be that structure that you need. uh, well, these are again conventional methods. We can also go for other forms of bioprinting, say, example, light based printing to create high resolution structures of such, so that this does not have to go through any sort of expensive methods of creating any dimension that you want or any structure that you require. So again, this is that case of microneedle patches being used. And obviously there are six subsequent amount of studies that they did, whether what is the difference between just using the microneedle versus using the microneedle plus using a microheater. So these are a lot of variables that you need to consider, and obviously the drug release is our main idea. So, doing histopathology and testing through the sections of the tissue that you require to check will help you confirm what is the amount of drug that has reached the site. So, following this, we have few cases, again, few more cases. And this is quite simple. This is fabrication of mucoadhesive oral films. You know that a lot of people have ulcers, or they have a tendency to bite their cheek and injure themselves, right? So the dentist might end up giving you a solution again, which is going to be placed at that site, and you have to keep applying it for, say, a week or more than that. So this is just a replacement to that, which is called a oral film, right? So it's a thin sheet of our hydrogel material. Here in this case, we are using hydroxypropyl methyl cellulose, right? And along with that, you have catechins which are used. Now, catechins are actually flavonoids. These are materials which are easily available, say in tea leaves and other antioxidant rich materials. So the main advantage of using such drugs, certain materials, are that they have an anti-cancer property, anti-hypertensive effect, and also anti-inflammatory effect. So when we are using HPMC, right, uh, we have to consider the concentration of, yeah, we have to consider the concentration of the HPMC, basically the hydrogel that we have used. And with that, we'll be able to understand what is the mechanical strength, or rather, what is the best fidelity that you could use for a oral film. Uh, if you notice, as I proceed through each of these cases, you have things to consider: mechanical strength, you have drug elution, zone of inhibition, then you have obviously your drug diffusion, maybe example heat diffusion. Then here we come down to the surface thickness, viscosity, polymerization, right? Few other factors that you need to see when you're printing something. So here they have tried to design multiple forms of uh, patches, or rather oral films, that could be used on a patient, in order to allow easier treatment or easier healing of any sort of ulcers. So viscosities are obviously something that you need to play around with, say 10%, 20%, 35%, and you optimize accordingly. So the porosity is another factor we need to check, to see whether there is a better drug dissolution, right? And the factor, an essential factors when you're using any sort of oral film or any sort of material, is also the surface area. So larger the surface area, more would be the amount of, or maybe rather the site of action. And also the thickness is an essential thing. So if you have a very thick structure, it will take too long for it to degrade. But if you're using a structure which is thin enough, or rather having an efficient thickness, it would also elute your drug, and it would also give you a sustainable amount of release. Again, thickness, as I mentioned, viscosity, polymerization factor, these are all something that they considered. So in this step, they try to create a basic um, film, or oral film. And I tell you, it seems very easy to create just a box like that, but actually it's not as easy, because when you're using a material like a hydrogel, you have to consider the gelation property, you have to consider the viscosity, the print, the efficiency and the efficacy of the print that you have created. And obviously we need to retain the structure we want to create. So after we have printed something, we need to see that it is cross-linked enough that my material is more stable. I can use this efficiently with all of these factors, considering the drug, the drug dissolution, the drug degradation, the surface area of contact, and so on and so forth. Um, from here, there's another final example, and obviously, I can give you some examples that can be quite easy for us to understand in case these were a little complex. So, like I said, formulation is one thing, right? So, now we are talking about something more complex. We are always starting with simple problems, and then we go progressing to more complex issues. So, a wound, a patch to a wound, a painkiller is one thing, and then now we are going towards anti-cancer drugs. So this study is actually the fabrication of 3D printed F-Gelma polymer, and again, it's local delivery. Most of our NDSs, NDDS that we have, their ideology is to create more targeted and more local drug delivery, right? So here they're trying to even create a more implantable structure. So whatever structures we have seen previously, these are all superficial, tropical sort of patches that you could use. But this is something which could be more implantable. So implants are something that have to go through a lot of vigorous testing in terms of the mechanical properties and the degradation factors, because there's a flow of blood through the body, there's a lot of movement in the body which can cause quite a lot of degradation. So here, in this case, I can be a little more elaborate. We are using doxorubicin. So doxorubicin is actually an anti-cancer medication, and it is used in cases of, say, lymphomas and sarcoma and any sort of cancer. But while it is a drug medication that is mostly used, it's also something which has a side effect to it, which is cardiotoxicity. So cardiotoxicity is basically the so whenever we talk about drugs, right, there's always an ill effect of a drug. Not always. In some cases, one drug will always, or in some cases, cause a lot of side effects or affect other parts of your body. That's where we can bring about our microfluidics or chip based studies, where you can study the effect of one particular drug on multiple parts of your body. So these drugs which you put, or rather use, they would have an ill effect, and to kind of reduce that, we have to go or sort to different forms. That's where doxorubicin, which is it's a material as such, or material in general, is now modified a little more to form something called a doxil. Doxil is basically a nanoparticle drug. So nanoparticles have this property of easier penetration, and obviously we have nanoparticle encapsulations. You have a shell and a core that you need to consider while creating a drug, and all of this is just to allow better efficacy, or rather for the drugs to stay on the site for long. Sometime, in some cases, you need a drug to stay on the site for long, and in some cases, you don't want it to stay for long. So here they have tried to create bioprinted hydrogel patches, and here you have used F-Gelma. So F-Gelma is actually fish gelatin based methacrylate material, and considering this material, conventionally Gelma is a high viscous material and it has a very sensitive gelation property. But when you're using this F-Gelma, which is an inexpensive form of Gelma, you will often notice a problem in the viscosity. So the viscosity is something that now we need to make smarter, right? So that is where you have CMCs being added, which is carboxymethyl cellulose. So we have created, or they have optimized and created a rather smart material, which is both viscosity wise, printability wise, and functionally also capable. And now they've created structures. The structures created are a cylinder, a, a torus structure, basically a donut like structure, and also a grid like structure. So grid lines are one, cylinders are one, and torus is one. So you use all of these materials, and we try to create different structures. And the idea here is to understand which shape is best as an implant, right? So we dealt with surface area, we dealt with the penetration of the drug, we dealt with the encapsulation of the drug, and now we're just talking about which is the best shape to consider. So there's a lot of studies that has to happen just before you just print a device. And 3D bioprinting is a tool which helps you create such structures more efficaciously, right? So to study the shape of the drug is what we're looking at. So obviously hydrogel concentration is one thing. So in this case, they have tested it with different sorts of uh, concentrations of F-Gelma. And we see there is a 10%, there's a 15%, and 20%. And as we see this, you'll see that 10% is the one which has the best swelling ratio. Swelling ratio is an important factor when it comes to hydrogels. So, based on the interaction with the solvent, the cross-linking density and hydrophilicity, these are also other essential factors we need to see. And in the end of the study, we understand or we understood that the torus structure was releasing too much drug, like a lot of drug at once. Then you have the grid line and you have the cylinder. So the cylinder structure is something which, again, while creating any structure, you have to remember that we have to go through a process of cross linking. So, when the cross linking does not happen efficiently, the structure is not most efficient when you're using it for that. So they noticed that cylinder was not the best in terms of structure fidelity, but in terms of drug release, it was great. So they followed using grid lines. Grid lines is a basic square with multiple layers and as a site of implant. So in the end of all of this, they were able to understand that a specific form of structure is able to create a better amount of drug release and better amount of implant sustenance mechanically as well as in terms of the drug action. So we I have told you or I have showed you about extrusion based printing where they have just created any form of structure. But now if you're trying to look at something like a drug eluting stent, or you're looking at a microneedle that you want to create using bioprinting as a technology, we also have our DLP based bioprinting. So here the advantages of using DLP bioprinting is fine channels, visibility, the resolution is great, and also the mechanical strength. So the kind of materials we use for printing these type of structures are actually supposed to be photopolymers or photoresins, which can easily cross-link and also create a good amount of mechanical strength. So it follows a basic SLA process. SLA is basically a stereolithography type of a printer, and the idea of it is to create more sustainable and stronger structures. So showing you a small video. Yeah, so this is a process where it prints layer by layer, and as it prints layer by layer, you have a very fine structure created. So it'll be wonderful in case of creating something like a microneedle, a drug eluting stent, in terms of creating more complex structures which require airtight porosity and a lot of calculations in creating fluidic forms. So with this, obviously there are a lot of products from our side. Incredible BioX6, BioX, and we have the BioMDX as well as Luminex. So these are the set of products that we have from our end. And then obviously we have BioVia, which is our other solution, which is meant for, which has one of its components, which is Discovery Studio. And this is meant for protein engineering, antibody modeling, simulations, anything from bioinformatics to cheminformatics. And I'm sure Divya has mentioned in detail about this to you in our previous session. And you will also learn more about this in our next session. Uh, so from there, now I come down to this bio quiz. And the question here is, what are the fields of research where bioprinting could work? So the options are genetic engineering, precision medicine, cell biology, or all of the above. Now you can select more than one options, and please note that the person who wins this is going to be someone who's correctly answered all of the 5 sessions that we've had. Right, so till now we've had like 2 questions, 3, 4, 5. So we'll have 5 questions. I will show this again in some time. You can go to our LinkedIn page, and also you can find us on every other social media platform where you can apply for this contest. Um, And thank you so much. Kindly follow us on LinkedIn and check out our products. We're always there to help you with any solution that you're looking for in your area of field, say mechanical or life sciences. And thank you. So, yeah, this is the end of this session. Just a second, I will present it.
Thank you, Aishwarya. That was a
Yeah.
very informative session. And I hope you guys all checked out the last bio quiz question that she posted. Do not forget to go and check out the LinkedIn page, and all of the instructions will be given. And as she said, you should have answered all the questions correctly. So far, many of you all are answering it in the comments. Please go to their page, check it out, follow the rules, and this competition will run for the other two sessions as well. And you stand a chance to win an Amazon prize. So I hope many of you are answering questions there and checking out Altum on LinkedIn. So I think we can have a Q&A session. So quickly
Yeah.
post in your questions. We don't have much time, so
Yes.
post your questions fast and we can start answering them. I think I saw a lot of comments saying, "Oh, aeronautics, I didn't know it was used there, and automotive." So see, you really don't know where and all it can be used. So don't keep this webinar to yourself. You can share it, and I can see a lot of people thanking you for your presentation. Many people even won the presentations, but yeah, it was very informative. Thank you so much. I think from last session onwards, it's going into the meat and potatoes of the concept. So I
Yeah.
hope
Yeah.
everyone's understanding. It's a really fun session, actually. Many people were shocked looking at the clients. They were like, "Oh, I didn't know these people had clients like this." So, see, guys, anything is possible. So don't think that you're just from the bio background. So this is fine. You can even share it with other fields. So it's interdisciplinary. So okay, I think we can start with the Q&A session.
Yes.
Okay, so we have okay. Okay, there's one question by Rishikesh which says, do we need any subscription to use BioVia Discovery Studio for our research work?
Okay. So, okay, BioVia Discovery Studio is actually a different product, like I said. It's a software, it comes in a pack, and it has a specific cost which you have to obviously buy and then use that product. So, subscription, I don't think subscription is the right word. You have to actually take the software, buy the software in order to use it. So, I hope this gives you a partial answer to your question.
Okay, there's one by, I'm sorry if I'm mispronouncing your name, Samir, I guess. I would like to ask, does the ink used in bioprinting have any side effects on cell viability?
Okay, so the materials that we're using for bioprinting, right? They are all supposed to be biocompatible, and they're all supposed to be cell compatible, right? So, in fact, the hydrogels or the bioinks we're using in most cases have to be able to sustain the cell's growth. Cell viability again is a very broad term to talk about, but the kind of effects our hydrogel can have on the hydrogel is on the cells is actually protecting it and also creating that ECM for it. So I wouldn't say hydrogels would have a negative effect on the cell viability. Maybe the printing pressure, maybe other factors will also come into picture when it comes to bioprinting.
Yeah, thank you. And okay, another question by Debjani. Sorry, Debjani. Yeah. Can 3D bioprinted hydrogel patches containing drug be used for neurodegenerative diseases in brain?
Okay, so I would say yes. There are a lot of applications that are available. People are working on multiple things. The three, four examples that I gave you are just something in general that was more easy for people to understand. I believe it could be used for degenerative diseases, except blood-brain barriers and these other things are something that you need to check on if you have to proceed with this step.
Okay, so mm, I think we can do another question, I guess, and then we'll be out of time. Uh, okay. Okay, there's this one question by Naga Prasanna. It says, if you're going to deliver the drug in muscles, for example, to treat inflammation, so can you do like a microneedle patch along with sensor and micro heater? So will it
Can
provide
you,
complete relief?
can you repeat that question for me?
It says, if you're going to deliver the drug in muscles to treat inflammation, so can you use microneedle patches along with the sensor and micro heater?
Yes.
Is it,
So
can it,
yes,
yeah.
yes. This the idea behind using this micro needle patch, which I mentioned about, is to create or is to deliver to the muscles itself. It's just allowing little better penetration over our general ointments or whatever we use. So, in short, the answer is yes.
Okay, so I think we can end the session there. It was very informative. Thank you, Aishwarya. It was so interesting.
You're welcome.
And many of the questions actually were answered as you progressed. So it was wonderful. Time flew by. And yeah, thank you. So, guys,
All
please
right.
don't forget to check out Altum on LinkedIn. Go follow and also share this webinar link. And also, you can watch this webinar again on YouTube. It is available. So, you all will have an idea as to what the future sessions will hold for you, and feedback link will be provided in the comments. So please don't forget, again, follow Altum on LinkedIn, follow Biotechnica. Thank you so much, and thank you,
Also,
Aishwarya. Yes,
yeah,
yes,
one more addition. So this what I've taken today is about basic drug delivery, and the next session that we have is going to be on drug discovery. So I think a lot of people who are in this field who are very interested would be excited to know that Dr. Divya is going to give you a lot of details on this. So please stay tuned and join us on the next session and obviously the subsequent sessions.
yeah.
So thank you so much. Yeah,
Thank you, everyone. Good night. Take care.
yeah. Good
Bye.
night. Bye.